So next I'm going to talk about AMX 114, which is our antisense oligonucleotide. I'll share some of our preclinical data, and then at the end of this presentation, I'll launch into our phase 1 first-in-human study design, which we call Lumina. Okay, so first, the scientific rationale for why we are targeting calpain-2 in ALS. So calpain-2 is a cysteine protease that's activated by calcium. It belongs to a family of non-lysosomal proteolytic enzymes. There are about 15 calpain isoforms, and they're really ubiquitously expressed throughout the body, with particularly high levels in the brain and the spinal cord. The two kind of predominant isoforms are calpain-1 and calpain-2. Calpain-1 tends to be activated by micromolar levels of calcium and is largely associated with neuroprotection, while calpain-2 tends to be activated by millimolar levels of calcium and has been implicated in axonal degeneration across a wide spectrum of neurological diseases. Both calpain-1 and calpain-2 are expressed highly in the brain and the spinal cord, and they are both very tightly regulated by the levels of intracellular calcium. Unlike other proteases that have been associated kind of with neurodegeneration, the calpains are highly selective in both the substrates that they cleave as well as kind of the locations that they cleave. And that becomes very important when we're talking about ALS, because some of the substrates of calpain-2 include a lot of different cytoskeleton proteins, including the spectrins, but also importantly, the neurofilament light chain, and then a number of microtubule-associated proteins like tau. Calpain-2 substrates also include TDP-43 and then a number of cell death signaling pathway proteins as well, including proteins involved in apoptosis. And I think it's also important to note here that there is an endogenous calpain inhibitor, which is calpastatin, which tightly regulates the activation of both calpain-1 and calpain-2. So partially because of these substrates, many of which we know are involved in the pathogenesis of ALS, as well as calpain-2's role in axonal degeneration and neurodegeneration, that was part of the reason why we initially got interested in this as a potential target. Calpain-2 sits kind of at this key spot in axonal degeneration. So as it gets activated by millimolar levels of calcium, it gets turned on. It leads to the cleavage of some of these key proteins. And then the next step is axonal degeneration, which then leads to motor neuron cell death. And there is a growing body of literature that supports why we should target calpain-2 in ALS, which I will get into in a little bit more detail in the next slides. So one of the first things that it got us interested in calpain-2 is that we know that calpain-2 levels are highly upregulated in individuals with ALS. So these are pieces of data from two different publications. The first is looking at the levels of calpain-2 mRNA from biopsied muscle samples of individuals living with ALS versus people who have no neurological diseases, the control samples here. And what we see is that the levels of calpain-2 mRNA are higher in some of the individuals with ALS compared to the healthy controls. In a separate study, investigators looked at the level of active calpain-2 in postmortem tissue from either motor cortex or spinal cord from individuals living with ALS, and saw that the levels of activated calpain-2, which is shown in the black bar here, is much higher in individuals with ALS compared to healthy controls. Whereas the levels of calpain-1, which again, we generally think of as being kind of neuroprotective, are unchanged between kind of the healthy controls and individuals with ALS. In animal studies, we have seen that calpain-2 inhibition has demonstrated benefit in an ALS mouse model. So this is a transgenic mouse model that overexpresses a human SOD1 mutation. And when that transgenic mouse model with the human SOD1 is crossed with a calpastatin transgenic mouse, so this is overexpressing the endogenous inhibitor of calpain-2, what you see is that there is an improvement in survival, as well as in motor neuron survival, and then also motor function. So here you have increase in the number of survival days, which you can also see here in the Kaplan-Meier plot. And then you also see that the time to disease onset in these animals is also progressed. In addition, when they did neuropathology on these mice, they saw that there was an improvement in the survival of the motor neurons, and the mice also improved motor function when they were running on the rotarod. And then finally, and I've already touched on this, we know that a number of kind of key proteins in ALS are known substrates of calpain-2. So importantly, there's calpain-specific TDP-43 cleavage products that have been detected in the spinal cords and motor cortexes of individuals with ALS and ALS FTD. And we also know that neurofilament light chain in particular is a substrate of calpain-2. So for these reasons, we developed our antisense oligonucleotide targeting calpain-2, which we call AMX 114. So to get a little bit more into detail about what AMX 114 is. So we thought that going after calpain-2 using an antisense oligonucleotide would allow us to selectively decrease the levels of calpain-2 in the CNS and not have any effect on calpain-1, other calpains. And so it really allows the selectivity over some other earlier small molecule-based approaches that have tried to go after calpain inhibition. So our antisense oligonucleotide is specifically designed to downregulate the expression of the calpain-2 gene. It targets an exon in the active site of the calpain-2 protease, so it doesn't lead to the decreases in calpain-1 or any other calpain proteins. And it lowers the level of the calpain-2 mRNA transcript through RNase H-mediated degradation. Going into the preclinical data now. So what we have shown in primarily iPSC-derived motor neurons is that treatment with AMX 114 preserves the health of axons and also improves survival. So this first set of data we did in collaboration with Charles River Labs. These are iPSC-derived motor neurons that then were exposed to different concentrations of a few different neurotoxic compounds. So we used vincristine and colchicine, both of which kind of go after microtubules and target axons. And then we also used rotenone as well. And so what we see here is that in kind of vehicle-treated motor neurons, after they have been treated with vincristine, rotenone, or colchicine, you get this release of neurofilament light chain out into the media, suggesting that there's been injury to the axons. And when these cells have been pre-treated with AMX 114, you get a reduction in the release of neurofilament out into the media. In collaboration with Sami Barmada, we also looked at an ALS-specific cell model. So here we are again using iPSC-derived motor neurons that are harboring one of the TDP-43 mutations, the M337V mutation. And in the panel on the left, what we are looking at is kind of the overall survival of these motor neurons over time. And so what you see is that the vehicle-treated motor neurons, these ALS model neurons shown here in red, over time, these cells start to die compared to healthy neurons. And in motor neurons that have been treated with AMX 114, you have this drastic reduction in the cumulative risk of death over time. In addition, what we saw is that similar to kind of the rotenone-treated motor neurons, we also have a reduction in the amount of extracellular neurofilament light chain that is being released into the media. So this suggests that what we're doing is, by inhibiting calpain-2 in these motor neurons, we're getting preservation of those axons and then also having prolonged survival in these ALS model neurons. And then finally, in collaboration with the Manfredi lab, we also looked at a different cell model of induced oxidative stress-induced cell death. So here again, we have a model of iPSC-derived motor neurons. These motor neurons were either treated with vehicle or AMX 114, and then they were exposed to 2 doses of hydrogen peroxide. And what happens in the control neurons shown here in black is that, within about 24 hours after being exposed to the hydrogen peroxide, you get this drastic drop-off in the cell body, and you get cell death, within about 120 hours or so. In our AMX 114 pre-treated motor neurons, what you see is that there is a higher percentage of the neuronal cell body that is remaining, and that this is prolonged out to 192 hours. We just have kind of an example here. So these are our control cells kind of before the treatment with hydrogen peroxide. Labeled in white are all of the cell bodies that you can see there. And kind of after treatment with hydrogen peroxide, you get almost a complete loss of the cell bodies. Whereas when the motor neurons are treated with AMX 114, you really have, in this example, near retention of all of the cell bodies. So on the basis of this data, we're really excited about the prospect of AMX 114, and we think that this is a good candidate to preserve the health of the axons and the motor neurons, and then preserve motor neuron survival as well. And so we're in the process of moving this forward into our first in human study, which, as I mentioned, is called the Lumina study. So at a high level, Lumina is a phase 1 multi-center randomized placebo-controlled multiple ascending dose study. We are looking to enroll approximately 48 adults who have ALS. And we will be giving kind of increasing levels of the study drug across 4 different dose cohorts. Individuals will be randomized 3-to-1 to either receive AMX 114 or placebo. The plan is to inject AMX 114 or placebo intrathecally, and participants would receive 1 dose approximately every 28 days for a total of 4 doses. Our objectives with the study, this is a first in human study, so kind of similar to some of the other studies that have been talked about today. Our primary objective is to understand the safety and tolerability of AMX 114. Our secondary objectives are to understand the pharmacokinetics of AMX 114 in both the CSF and the plasma. And then our exploratory objectives are to understand the effects of AMX 114 on key biomarkers in both the CSF and the plasma, as well as functional measures of ALS disease progression. Our key trial entry criteria are here. We are looking to enroll adults who are either have clinically definite or clinically probable ALS based on the ALS-Coriell criteria. And the time since onset of first symptom must be less than or equal to 24 months. Importantly, we are looking for individuals who have a slow vital capacity at screening of greater than or equal to 75%. And then they can be on approved treatments for ALS so long as they're on a stable dose for at least 30 days prior to the baseline visit. This is kind of a schematic of our study design. So there'll be a screening period of up to 4 weeks. Then individuals will enter into the treatment period, which will last for 13 weeks. And then we'll go into a safety follow-up period for about 8 weeks. During the treatment period, they'll receive intrathecal boluses of either AMX 114 or placebo every 4 weeks for a total of 4 doses per cohort. And we are planning to start at the lowest dose cohort with a dose of 12.5 milligrams, with individuals randomized either onto AMX 114, 9 people, or placebo with 3. And then importantly, we are planning on having an open label extension once we've had the opportunity to evaluate the safety and the efficacy data to make sure that there's a positive benefit risk profile. Here's a more detailed look at our endpoints. So our primary endpoints, again, are focused primarily on safety and tolerability. Our secondary endpoints are really focused on the PK. And for our tertiary endpoints, this is where we really want to understand the pharmacodynamic effect and look at the biomarkers. So I think especially given what we know about calpain-2 and axonal degeneration, we are looking at some of those key cytoskeletal proteins for a readout of proof of concept here. So we'll be looking at calpain-2 levels. We'll also be looking at neurofilaments, both neurofilament light chain as well as neurofilament heavy. And then we'll also be looking at some of the spectrin breakdown derived products as well, because we know that those are kind of direct substrates of calpain-2. We'll also be looking at ALS progression measures, including the ALS-FRS-R and slow vital capacity. This is my last slide. So in summary, we are very excited about targeting calpain-2. We think this is a critical effector of axonal degeneration and is a key early contributor in the pathogenesis of ALS. We've developed AMX 114, which is an antisense oligonucleotide inhibitor of calpain-2, which is highly potent and highly selective. We have seen benefit in our preclinical models on both neuronal survival as well as on axonal health through a reduction in neurofilament light chain. And we are going to be initiating our phase 1 study of AMX 114 in adults living with ALS either at the end of this year or in early 2025. And I think with that, I will pause and take questions.
Thank you, Lauren, for sharing that great update and the rationale for moving forward. We have a couple questions. The first two I actually think are related, or at least to me they are. What's the half-life of calpain-2?
Yeah, so when we look in our animal models, it's relatively long-lived in the CSF. You know, we're giving it directly into the CSF compartment. Some of it does kind of spill over into the periphery. So the calpain-2,
Yeah,
not
the
AMX 14.
target, but that would be a good question to answer as well.
All right, I'm happy to answer both of them.
Yeah.
So calpain-2, you know, it is expressed throughout the neuron and the cell tissue that it's in. The half-life seems to be, there's not a lot of studies on this, but from the literature, it seems to be kind of on the order of like days to maybe sort of early weeks, which coincidentally is also the half-life of our drug in the CSF. We see that the half-life in our animals is about, you know, on the order of days to weeks.
And do you have a sense of how much knockdown you expect to need to see in order to prevent cleavage of TDP-43 or NFL or other substrates?
It's a really hard question. I think it's hard to know because unlike a lot of the other ASOs that, you know, like tofersen or some of the other ASOs that are in development where, you know, we're trying to knock things down by as much as possible. I think it's a lot harder to know for calpain-2. It's a little bit of a different kind of a target that we're going after here. So I think we will learn a lot on the basis of kind of as we're getting our pharmacokinetics and pharmacodynamics from the first in human study. I think on the basis of our preclinical data, we have reason to think that even, you know, a little bit of knockdown of calpain-2 seems to have benefit in terms of the amount of NFL reduction that we're seeing and also the survival. But I think it's very tricky to kind of make that translation over to what that actually means for people.
And I do have a couple of questions related to the preclinical work. So
Yeah.
we've had a few sort of gene therapies and ASOs being described today, and a lot of work has been done to confirm the specificity. Did you do a lot of that work as well? And do you have a sense of the specificity of your drug?
Yeah, so we did probably similar to a lot of the other compounds as well. We did a lot of in silico modeling as you know, as we were designing the ASO, and then, you know, initially kind of screened a wide panels of ASOs, you know, to pick the one that is both the most potent as well as the most specific. So yes, we in general have seen that it is highly specific for calpain-2. Importantly, we don't see knockdown in kind of any of the other calpains as well. And based on our in silico modeling as well, we know that like there really are no other genes in the genome that should be knocked down on the basis of this. So.
And then one other question, so some of the preclinical studies you did vehicle versus your drug. Did you consider, or do you have any studies where you also explored like a scramble ASO as one of your controls?
It's a good question. I would need to double check with our preclinical team so I can get back to you on that. I think in general, we mostly did the vehicle and didn't do as many studies with the scramble ASO, but some of the early ones might have been with scramble as well.
And we have one more question in the chat and then we'll move on. Do you plan to measure levels of YKL-40 and TDP-43?
Yeah, we are really casting a wide net with the biomarkers that we're planning to look at in this study because I think we are going to learn a lot about, you know, calpain and axonal biology. I will say, yes, both YKL-40 and TDP-43 are on the list of markers that we're planning on looking at.
Fantastic. Thank you so much for your presentation. Appreciate it.
Yeah, thanks so much for letting me be here.